You can start, ma'am.
Okay, okay. Yeah. So, good morning to one and all. Like, it is an immense pleasure on my behalf to be a coordinator of such an uh, important and very informative and useful series of lecture uh, for people who have done and completed their PhD and will be the guiding torch for the next one students to follow up. So Deshmukh sir has given me this opportunity. So I am really grateful to sir in particular and to the society as well for providing me the platform. Now, today's speaker we have today. This is the starting session for the 2024 series. So we have as the opening speaker Dr. Moumita Sharkar. Moumita has been known to me for more than a decade, and a very good morning, Moumita, and wish everyone, each one of you present here, also a very good morning. So Moumita has completed her BS from Ashutosh College, Kolkata, and then in biochemistry, and followed by biochemistry MS from University of Calcutta. Then, after qualifying the NET exam, she did her PhD in the Institute of Indian Institute of Chemical Biology under the supervision of Dr. Mrinal K. Ghosh. In 2016, she completed it, and her topic was studying RNA helicases in cancer. Then she joined Professor Neil Dillon's lab at MRC LMS Imperial College London, where she studied the roles of RNA binding proteins in embryonic stem cell differentiation. Currently, she works as a research associate with Professor Costanza Emanueli in NHLI Imperial College London to unravel the mechanisms of novel class of small RNAs in cardiovascular diseases. So this much for Momita now, and I would request Momita to start on with her. Yes. Good
Thank
morning,
you, ma'am.
Halami sir. Just I didn't wish you in particular, but you are regular attendee of all lectures. So
Thank you very much.
welcome. Yeah.
Very interesting topic, and the speaker is from IICB, I believe, so it will be a pleasure to listen her.
Yeah. Okay. Yeah. So the platform is yours. Yes, Momita.
Good morning, everyone, and thank you, ma'am, for the kind introduction, and thank you for providing me the opportunity to present here. And thanks to all of you for coming for this lecture. So I'm sharing my screen. Please let me know that if everything is okay technically. You can't share my screen, right? See my screen, right?
No.
Okay, give me a moment. I'll try to share my screen. Somehow I can't share my screen. Give me a moment so that I can sort this out. Sorry about this.
About you
Yeah, I
and,
can't.
and then rejoin. Sometimes it happens, or like if you then wish to send it to me so that I will
Yeah,
look.
maybe I can do that. I'll log out and rejoin. If I can't, then I can send the presentation to you. Give me 5 minutes. I should be back. Sorry about this.
So is the screen visible to all now?
Yes, so you can see the screen.
Okay. Momita, you may start. I think she has joined back or no. Let me write to. Yeah, it is visible. You join back. Okay. Hello. Yeah. Yeah, I am. I have already shared the screen. You just join back. Huh.
Thank you, ma'am, for sharing the screen.
Yes. Okay. Hope it moves again. Yeah.
I hope my voice is clear. So thank you again, and sorry for the technical glitches. So today I will be talking about quite a bit of diverse topics. So I'll be talking about gene regulation in pathogenesis by a novel class of small RNAs. And next slide, please. So just briefly, as ma'am already introduced me, I would just briefly run through. So I did my biochemistry bachelor's from Ashutosh College, moving to master's in biochemistry, and then I did my PhD in cancer biology from CSIR IICB. Then in September 2016, I moved to London Institute of Medical Sciences for my postdoctoral research, and currently I work in National Heart and Lung Institute, Imperial College, London. Next, please. So yeah, this is a very funny way to start the presentation. So as you see, being a molecular biologist, there are different memes which you see in Facebook and in Twitter and Instagram, like when a general person sees. But I guess we scientists and molecular biologists do not really represent any of these pictures which are here. Of course, in a day, we don't change people's lives. But yeah, what you say, droplets of water eventually forms a sea, make some difference in people's lives. Next, please. So just a snapshot, how important molecular biology and where we stand. So yeah, we have the chromosomes and the DNA, which gets a very basic central dogma, which gets coded into RNA and protein. And so the three most important events of gene regulation in a cell is, of course, transcription, replication, transcription, and translation, as we always know. And of course, here the focus of today's topic is more the RNA. So we will be stressing more on different kinds of RNA and its roles. Next, please. So just to brief through just quick basics, which because most of us are not familiar with different types of RNAs, like because there's too many stuffs. So of course, messenger RNA is the most well-known, the most abundant, which is translated into the proteins for protein synthesis. Then of course, we have the ribosomal RNAs, the transfer RNAs, which we all know, which we all are familiar with. Then the heterogeneous nuclear RNA. And then there has been recent discoveries, not recent, I would say, in the late 90s when the microRNAs and the small RNAs were discovered, and which were not coding, like it don't synthesize into proteins, but what it does, it has regulatory roles, and it impacts gene regulation in a very critical way, which is important for different functions of the cell. Next, please. Next slide, please.
Somehow it got stuck. I am trying. I don't know why. It was moving fine. What happened? So much of problem. Yeah.
Thank you. Sorry.
Fine. Yeah. Yeah.
Okay. So for today, first of all, I would be talking about my PhD work, which is RNA helicase P68 in colon cancers. There will be very diverse topics. Then we'll be moving into discussing about roles of RNA binding proteins, focusing on C03 in stem cell development. And finally, I would also be talking about small RNAs and its roles in diabetes and ischemic heart disease. Okay, next slide, please. So for my doctoral work, which I started with Professor Mrinal K. Ghosh, who is currently the head of Cancer Biology and Inflammatory Disorder Division in CSIR IICB. And my work focused on role of RNA helicase P68 in cancers. Next slide, please. So basically, just to give all of you a snapshot of what RNA binding proteins are. So of course, RNA plays different roles, but RNA binding proteins are critical in modifying different roles of RNA. Some proteins bind to RNA for RNA export for different functions. Some proteins bind to RNAs for alternative splicing so that it can lead to different RNA products and different proteins. Some binds to RNA to regulate the translation into protein synthesis. Some helps and binds in RNA degradation, while others help and stabilizes the RNA. So there's diverse roles which are in a snapshot here of RNA binding proteins. Next slide, please.
I don't know why it is happening. It's working and again non-working.
Hmm.
Is it okay?
Yes, it's okay. So here we see cancer, which the very famous author Siddhartha Mukherjee wrote about it, The Emperor of All Maladies, the biography of cancers. And of course, cancer research, we all know, has cancers, how it affects worldwide, and how there are severe forms of different variety of cancers, some of which are still pretty uncurable, though there has been a couple of trials. Some are getting better, like breast cancer, cervical cancer, vaccinations. So yeah, cancer research is definitely very, very important because it is so diverse. It has so many bypasses. So let's try to understand one part of it. Next slide, please.
Momita, it's bad luck. Why each and every time it's getting stuck? I don't know. I'm not doing any other functioning. I'm not doing any other function, but only doing this, but still.
It's a bit slow, I think, maybe because of the internet.
That could be. Just a minute.
घंटा लेट खबर उठेगा माने 7 बजे की जगह 8 बजे को
Okay, now here we have the next slide. So yeah, I think it's because of the internet probably. So here we have
At the right slide now?
Yeah, we skipped one slide, but that's all right.
Okay.
Yeah, that's the thing. So, but it was very short. So as you see that when cancer cells proliferate, how it can be different from normal cells. So cancer cells multiplying and invading in the picture. And often it is hyperactivated and aberrantly regulated at different levels like proteins, genes. Everything is totally deregulated, which leads to cancer cell activation, invasion, proliferation. Next slide, please. So this is a very, very famous hallmarks of cancers. People working in cancer research, when we start the cancer research, the very basic things which we read is the hallmarks of nature reviews of hallmarks of cancer. So hallmarks of cancers are basically few of these properties which you can see on the screen. One is sustained proliferative signaling. So the cancer cells are hyperactive. It keeps on, proliferating, and for that it needs growth signals which are hyperactive. Then resistance to cell death. So in a normal homeostatic condition, there is a balance between cell growth and cell death. So if there is not enough cell death, then there is accumulation of dead cells, accumulation of aberrant cells. So when a cell is cancerous, there is resistance of cell death as well, leading to more proliferation of the cells. Then induction of angiogenesis. So angiogenesis is a term which we say is increased in blood vessels. So increased blood vessel formation, because when there is high number of cell growth, you need high amount of nutrients and high amount of blood vessels for the transfer of the nutrients. So therefore, when the cancer initiates, there is increased angiogenesis, that is blood vessel formation. Then, of course, increased replication of the cell. And then finally, of course, inhibition of tumor suppressors, that the factors in the cells which are tumor suppressive, there is inhibition of those factors. And finally, when the cancer passes some of the stages, like later stage, it starts metastasis. So metastasis is the term so basically at the initial stage, cancers are more primary. So it is localized to one particular site. In a more advanced stage, these cells started dislocating from that particular site and start spreading into different parts of the body. And that process is called metastasis, which is majorly observed in high-grade cancers and can often be very, very lethal and causes multi-organ failure. And it's always very, very wise if the cancers are detected in advance, before it starts metastasizing in the patient's body. Next slide, please. Okay, so why I started my PhD in the first place was when I was studying biochemistry in University of Calcutta. We were taught as a part of our course curriculum. I'm sure you guys also learned that is cell signaling. So I was extremely fascinated by cell signaling, and I wanted to pursue research. I was so fascinated by cellular signaling and the gene regulation. I wanted to pursue research in cell signaling, and I wanted to go to America for doing my PhD, which I ended up, but for some personal reasons, I didn't go. And that point of time, my sir, Mrinal K. Ghosh, he just returned from America, and he did exact cancer signaling research. And I was so fascinated that I ended up joining his group. So basically, it's cancer signaling or any general signaling in the cell is very analogous to, if you imagine a city where all the in a traffic system, the signals are all fine and the city is running smooth with the traffic is fantastic. And imagine just a signal failure at any point in a particular point in a city. Say, imagine there is a signal failure at Goria Hat or a signal failure at Park Street, and the whole traffic gets messed up. This exactly happens to the microsystem as well. This exactly happens to the cell. When there is a particular aberration in any point, any important factor, the whole gene regulation of the cell gets messed up, and which causes aberrant signaling, aberrant gene regulation, leading to different sorts of pathogenesis, including cancer. Next slide, please. So in terms of general, just to give you like a general snapshot of signaling pathways. So what happens is there's of course a stimulus, a growth stimulus or whatever stimulus. There's a receptor which is present on the cell surface, which then receives the stimulus. That is, the stimulus or the ligand binds to the receptor. Then there are a series of proteins which relays the signal into the cell. That's why it's always called a signal transduction cascade or a signaling cascade, which leads to manifold amplification of the signal. And then eventually, after a series of relays by several factors, it hits some of the critical regulators called transcription factors, which then goes into the nucleus and then alters the gene regulation of different aspects of gene regulation and turn on the genes, which ultimately cause phenotype of the cells. In terms of cancers, I mean, there are numerous signaling pathways which are very, very important for cell growth, cell metabolism, and different aspects of cancers. But few of the proteins I'm mentioning here are very, very important. There has been clinical trials and different kinds of drugs targeting these receptors. For example, you can see here, one is VEGF, FGF. VEGF is vascular endothelial growth factor, which is very important for angiogenesis, that is induction of blood vessels. You see platelet-derived growth factor, that is PDGFR. You see EGFR and IGFR, which is epidermal growth factor and insulin growth factor. And also you see WNT, WNT, which was also initially identified to be very important molecule for signaling, and especially which is often deregulated in cancer cells and cancer signaling.
Zoom meeting चालू.
Next slide, please.
PowerPoint presentation.
So
IICD
when I
मध्ये.
joined Dr. Milal Ghosh's lab, that point in IICB, we were a fresh team of 10 PhD students and very enthusiastic. We focused on very important signaling pathways in cancer, especially EGF signaling and Wnt signaling. And we were mainly focused on regulation of these signaling pathways at different aspects. Like some of us focused on how these pathways are, how the proteins of these pathways are degraded or saved. And some of us focused more on how these pathways are regulated by different other genes and transcription factors. And in a whole, we all were contributing towards how these critical pathways are balanced in cell proliferation, growth, and cancers. Next slide, please. So I was working with P68. So it is an RNA helicase. So helicase is like what it does. It unwinds the RNA and helps in different kinds of functions. It's a very important player of RNA metabolism. It plays several roles in canonical RNA metabolism, like synthesis of RNAs, biogenesis of RNAs, and splicing, and different other aspects of RNAs. It's very important, very, very important for RNA metabolism and RNA synthesis. But then there were reports that apart from functioning in RNA metabolism, it was found to be overexpressed in wide variety of cancers. So overexpressed is like it was more, much, much more. Its levels were much, much more in different types of cancers. And it was also found that it was a co-activator of different transcription factors. So as I was mentioning you, transcription factors are critical factors, which are pretty downstream of the cell signaling pathways. But these are the factors which bind to the genes and then activates or inhibits the transcription of the genes and hence the phenotype of the cell. So this RNA helicase P68 was a co-activator. So what it does, it binds to these kind of transcription factors like beta-catenin, which was a transcription factor of Wnt signaling pathway, or estrogen receptor, which is a transcription factor of estrogen signaling important for breast cancer, and then androgen receptor for prostate cancer, NF-kappa B for prostate cancer. So these different kind of transcription factors, and this particular protein was a co-activator of these transcription factors, and it was already known. So we were very interested in knowing more about this RNA helicase P68 and how it functions in cancers and how itself it is regulated in cancers. Next slide, please. So for me, in my PhD, I deciphered or unraveled P68's roles in two different pathways, very important for cancer cell. So one of them was AKT pathway. So AKT is a central kinase. Most of you must be aware of it. It is very, very important for cell protein translation, cell growth, cell metabolism, cell cycle. Without AKT, the cell would die. Absolutely, absolutely die. And then AKT also regulates a factor called FOXO. So what it does is there is a homeostasis in normal cell. So in normal cell, FOXOs checks the function of AKT. So as I was mentioning you, homeostasis is very important for normal functioning of a cell. So when AKT gets overactive, FOXOs then checks its function that, so that it doesn't synthesize more proteins. So FOXOs are very important tumor suppressors, which were discovered in late 80s. So AKT is oncogene, which is important for cancer, and FOXO is a tumor suppressor. So the balance is very important in normal cells. In cancer cells, what happens is AKT phosphorylates FOXOs and degrades FOXOs so that the FOXOs totally goes inactive and AKT completely takes over the cell to cause cancer. Next slide, please. So here I'll show you just some of my results, which I obtained that time in my PhD. So as you see, this is in the left, you see this is very flowery structure. So these are the samples we obtained from PG hospital, and these are the colon cancer and the normal colon samples. The flowery, very nice organized structure is definitely the normal colon. And this is a it's a histochemistry where we stain the sections. So we take the blocks from the hospital, then we cut the sections, and then we check the protein levels by performing immunohistochemistry. That is, you stain the particular section of the tissue with particular antibody, with particular protein, and then you add an antibody you're interested in, and you see how is that protein affected in the tissues. So here, first of all, you can see the morphology of the cancer sections, which is completely distorted compared to a beautiful organized flowery structure in the normals. And then, of course, our protein of interest was RNA helicase P68. So you see that P68 was very, very dark compared and very, very highly overexpressed compared to the normal counterpart. AKT, of course, was supposed to be, because it is known to be an oncogene overexpressed in cancers. What we were trying to understand is the role of P68, and we saw we screened number of cancer tissues, and we observed that P68 and AKT was exhibiting a positive correlation in terms of expression in the cancer tissues. Next slide, please. Here you see, so what we these are no Western blot images. Just you isolate the protein from the cancer cells, and then you probe with your antibody of interest, and you see. So what we did was we designed, we knocked, so we silenced P68. So we inhibited the function of P68 by using a reagent which we call siRNAs. So they basically inhibit a function of a protein. So to see that what effect it has on AKT, because in the literature, it was already known that AKT was regulated by Wnt signaling factor beta-catenin. And then in other literature, it was known that P68 is a coactivator of beta-catenin. So we were trying to match 1 + 1. So okay, P68 helps beta-catenin. Also, beta-catenin is known to help AKT. So is there a relation, direct relation of P68 directly controlling AKT? That's what we were trying to decipher in the whole project. And indeed, we did see this. So when we were silencing P68, we could see decrease in AKT levels. Here, this is the RNA levels, which we did by quantitative real-time PCR. And you see the same results, that when we were silencing P68, there is a significant decrease in the AKT mRNA levels. Then we performed, so we screened several, so then we were screening the AKT promoter region. The promoter region of a gene is the region which is important for its transcription, and different transcription factors bind to that region. So we found that there are several beta-catenin binding sites and also NF-kappa beta binding sites. And we deleted those binding sites here, which you can see in the bottom left panel. And definitely you see that P68 was regulating through these binding sites. And then we performed chromatin immunoprecipitation, where we pull the whole chromatin of the cell. And then we try to see whether the particular factor is bound, which is very popular. It's called CHIP assays. And we did find that P68 occupies the promoter region of the AKT gene and helps via beta-catenin, indirectly occupying the promoter of the AKT gene and regulating its transcription. Next slide, please. Then we generated, we thought of replicating our hypothesis in an in vivo model. So we had, in the mouse model, we had a parallel cell line, which we call a syngeneic cell line, which shouldn't elicit any immune response in the mouse, which was syngeneic for the colon cancers. So what we did, we made a cell line which was supposed to overexpress P68. So as you can see, when we injected these cells into the flank region of the mouse, and after 21 days, we observed that the tumor volume in the mouse was about 8 times more. So this was first time we were reporting something like this of our P68 overexpression. And then we did all the staining and the pathological characterizations. And we did find that increase in P68 increased AKT and decreased FOXO3A. And So the whole paper, next slide, please. So the whole project and the whole paper dealt in the summary as that P68 co-activates, that it helps transcription factor beta-catenin and transcription factor NF-kappa B, indirectly increasing the transcription of AKT, which then inhibited the tumor suppressor FOXO3A, and this helps in cancer progression, especially colon cancer. This was published in Oncogene, and we were very happy. This was my first publication. And next slide, please. In the second part of my PhD, so when I was doing this work, STAT signaling was one of our very important signaling pathway, because we used to study a lot of EGF signaling. So STAT signaling is another signaling which was very important to play important roles in brain cancers, glioma, breast cancers. But its roles in mechanisms in colon cancer was not very well studied that time. So I took the opportunity to think that, well, maybe P68 could play some roles with STAT because it is playing roles with beta-catenin. So here's a snapshot of STATs. So similarly, when there's a receptor of STATs on the cell, and when it binds to the growth ligand, STAT3 gets phosphorylated because it itself is a very famous transcription factor. It goes into the nucleus, and then it upregulates the gene expression of several proteins which are pro-survival, that is, we increase the cell proliferation, cell angiogenesis, and when aberrantly activated, causes cancers in different types of tissues. Next slide, please. So as you can see, we did similar experiments where you see there is a beautiful normal colon and a very bad morphology and overexpressed P68 and STATs in colon cancers, which exhibited positive correlation. Next slide. And then here you see is a very classical experiment. It's called immunoprecipitation, where you use antibody and you see whether one protein binds to another protein. So we screened several colon cancer cell lines, and we found that P68 indeed directly binds to STAT3 protein. And the right is a technique called proximity ligation assay. It's not a very famous technique, but it's a very, very cool and a very nice technique. So here you can directly visualize the interaction between two proteins in the cell. So if you see the red dots indicate interaction between two proteins. So there are two proteins which are labeled by two fluorophores, and when they are close, that is when they are interacting, there is a signal in forms of a color which you can quantify and visualize where it is particularly located in the cell. And if there is no interaction, then you wouldn't see any signal or any color. And here it's just one sample image where I did see there is a very strong interaction of P60 and STAT3 in the colon cancer cells as well, especially in the nucleus, because these are blue, which is stained by DAPI, which indicates nucleus of a cell. Next slide, please. And then, of course, we did the silencing of P68. And as you can see, there is a panel of genes like MCL1, BCLXL, VGF, all are STAT3 target genes, which all are downregulated. And chip assays confirm that P68 binds directly to all the promoters of the genes along with STAT3. So we came to the conclusion that P68 again co-activates STAT3 mediated gene expression in colon cancer cells. Next slide. So again, the conclusion of this study was P68 co-activates STAT3. So it was very, very, very nice study, like where I identified a novel co-activator of STAT3 being such a famous transcription factor in oncogene, and which causes oncogenesis. And this again was published in Oncogene. And I was very, very happy to discover P68 as player of two very important signaling pathways in normal cells and in cancer cells. Next slide, please. So yeah, this was the summary, and I was really, really happy in my PhD lab. This you can see Malini ma'am here in the left picture. This was picture from 2010 or 11, and our whole group that time. And this is a recent picture when I went to India 2 months back. So I'm very happy to see everybody very successful, doing very well. And I learned a lot, like learned everything from the scratch, all the techniques. And I think I learned a lot from my PhD lab and a lot of independent thinking, which is very important, coordination and execution of the projects, which really helped me further down the line. Next slide, please. So then there was a point when I was applying to my postdoctoral position in several positions in America. I was really not keen on going to Europe because I didn't know many people. But well, destiny took me to London. And next slide, please. So then here for my postdoctoral work, I joined Professor Neil Dillon's group in September 2016 in UKRI, London Institute of Medical Sciences, Imperial College London. And the work which I was doing was role of C03 in stem cells and development. So we will talk now a bit more about stem cells and development as well, which will be of, I hope I will make it interactive because there are various topics to absorb. Next slide, please. So C03. This is again another RNA binding protein like P68. It's not a helicase, but this is complex of CCR4 not complex, which is the most important deadenylase. That is, it degrades all the mRNA of the eukaryotic cells. So because mRNA needs to be degraded because of the normal turnover of the cell, so this is the particular complex, and C03 is not the enzyme, but it is one of the important subunit of this whole complex, which aids in mRNA degradation. Next slide, please. So just to give a brief shot of some of the stages of embryonic development. When I started my postdoc, I was clueless about these things. Like last I read in school, I'm like, okay, oh, what's this now? So anyway, but then it's really interesting and very, very important in terms of cancers, development, stem cell regeneration, and therapy. So here you see, it's a very brief and a nice snapshot. So when the egg is fertilized, first in 2.5 days, in mammalian development, it forms an eight cell stage. Then there is a blastocyst stage. So blastocyst stage is a very like a 4.5 days, a very early cyst stage where there is no still segregation and still like a round mass. And then at 5.5 day, there is implantation of the embryo. And during this stage, some of the initial layers, which are the markers of different lineages forming in the organism, is a very important. It is called a primitive streak structure, which is called mesendoderm, which then differentiates into three, marks the first stage of differentiation of three layers. That is endoderm, mesoderm, and ectoderm. Without this, we wouldn't exist. So later on, endoderm leads to lungs, thyroids, and the digestive system. Mesoderm leads to cardiac, heart, skeletal muscles, red blood cells, and smooth muscles. And ectoderm forms the skin and the nervous system. So this stage, that is a very early stage of the primitive streak and the mesendoderm, is very, very important for development of all organisms. Next slide, please. Now, stem cell. So I really, I was very, 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 what you say, very, very interested in cancers. And at the beginning, I was like, okay, I have a cancer project and I have this stem cell project. Am I even interested in this? But slowly, I grew very, very strong interest in stem cells as well. So here you see, apart from the canonical mammalian development study, why the stem cells are so important currently. So as I was telling you, after this fertilization of the sperm and this blastocyst stage, so in the very early blastocyst stage, whether it's a mouse fetus or the human fetus, you can harvest, a researcher can harvest some of the cells which are called the pluripotent stem cells. Now, these cells are pluripotent. They can generate and differentiate into any lineage you want. And these are actually the cells which then gives rise to, so differentiation, so pluripotent means it can differentiate into any cell type. So as the cell progresses further, it starts losing the potency. That is, from pluripotent, it becomes slowly more differentiated, is more committed to one lineage, it starts losing the pluripotency. Now, nowadays, it's a very hot field of research is regenerative medicine. So now the stem cell field is so well studied in terms of pluripotency, in terms of the pluripotency factors. So now we often know that when there is a problem in the body, and then when we put a graft in the patient's body, then there is immune response and graft rejection. So that's how stem cell therapy is so important, because now the stem cells can, so if suppose it's something is called induced pluripotent stem cells. So now from a differentiated cell, you can reverse program the cells to the pluripotency cells, so that you can generate fresh cells and help a patient. For example, if you have a skin biopsy, if you see the top right image from a patient, and then you treat those cells with reprogramming factors, and you can actually reverse program the cells to generate pluripotent cells. And then from those pluripotent cells, you can generate fresh, like any tissue of the patient, like cardiac cells, gut cells, blood cells. And you can also study, test drugs, model what is, because now it's the era of personalized medicine. My medicine might be different from yours because of different genetic composition, and also the elicitation of immune response. So that is how this IPS cells, that is the induced pluripotent cells, and the reprogramming is so important nowadays for personalized therapy and personalized stem cell therapy. And it's very, very critical to understand. People publish like very, very good papers, and very good research is going on in this field. Next slide, please. So back to what I was doing. So this particular RNA binding protein, which was C03, which was already known by several very established papers from very big groups of the world, that it was already involved in pluripotency of the cell. So it occupies, like, apart from its, like P68, similar like P68, apart from its role in canonical RNA metabolism, it was also important in maintaining the pluripotency of the stem cells. It was also involved in cancers. So our question in our group, we were asking, okay, we know that it is important in maintaining the pluripotency of the cell, but what about its functions when it goes for lineage commitment, that is differentiation? Next slide, please. So at that point, Professor Neil Dillon's group was working with another protein called Aurora B. So this particular protein is a kinase. So what it does, it phosphorylates. So kinase is a protein which will always phosphorylate another protein to alter its function. So this was a particular protein which is very important in cell cycle and mitosis. Without this, cell cycle would stop. So important for cell cycle, mitosis, cytokinesis, and different stages of cell cycle. But apart from, again, the canonical roles, we were trying to identify what are the novel roles of Aurora B with C03. Next slide, please. So what we identified is that, again, by immunoprecipitation reaction, that Aurora B directly interacts with C03. So this we had one information, that Aurora B interacts with C03. Okay, fine. Now, by mass spectrometry, the lab had identified that Aurora B phosphorylates C03 at two particular residues near the nuclear localization signal. So, each protein has an NLS, that is nuclear localization signal, which gets activated, and then it leads the protein to go into the nucleus. So that's what Aurora B was doing to C03. So we were trying to see whether it is true. So you can see below in this picture. So we identified the sites where Aurora B was phosphorylating C03, and we mutated that site, we altered that site. And by altering the site, we did see that the nuclear localization was completely abolished, which we inferred that it was indeed very important for the Aurora B to phosphorylate C03 and send it to the nucleus. So by now we have, okay, Aurora B interacts and phosphorylates C03, so what? And send it to the nucleus. Okay, then what? Next slide, please. So now we were asking the question in the context of stem cells and differentiation. So, okay, Aurora B sends C03 to the nucleus. Does it have any function in terms of stem cell pluripotency and lineage commitment? So we have a technique as a stem cell biologist. So what we do is a very gross technique. It's mimicking an embryo in a very gross way into the dish, which we call as embryonic body. Of course, it is very, very gross. It's not like a proper depiction, but you can understand. So what we do is we collect stem cells, we give some factors, we take off the pluripotency factors, culture it in a particular way, and it forms embryonic body, which contains 3 layers, that is endoderm, ectoderm, mesoderm. And you can identify if it is forming properly the initial layers for further development. So you see here, so here brachyury in red marks the mesoderm. FOXA2, these are very popular markers. FOXA2 marks the endoderm. And we were trying to assess that whether the mutation of the particular C03 by Aurora B causes any effect. So when we plated the mutated cells, you see that it was not forming these lineages properly, unlike the upper panel, which is the normal cells. But the ectoderm here in the right were fine. So we deciphered that this function, that is Aurora B phosphorylating C03 and sending it to the nucleus, has some roles in helping the stem cells forming mesoderm and endoderm. Because when we were mutating it, we can't see the effect. Next slide, please. So then we tried to very intricately understand what are the roles, because you have to really, really fine tune it. So I was doing differentiation in a very particular way. So we studied a couple of factors. So how mesendoderm, that is mesoderm and endoderm, forms. So basically, these 4 signaling pathways are very important. One is Wnt pathway, which we talked in the cancer part. Another is BMP pathway. Another is FGF, which is fibroblast growth factor pathway. Another is activin pathway. So we were modulating and adding each particular pathway and trying to see which particular pathway is involved in the C03 mode of mesendoderm formation. Next slide. So here you see these are the differentiated cells from the stem cells. So we see that what we were adding BMP and FGF and trying to induce differentiation into mesendoderm. And again, we saw that the mutated cells below couldn't form really both the layers, that is the mesoderm and the endoderm. And we inferred from the embryonic bodies and also from integrate differentiation of the embryonic stem cells that these 2 particular germ layers at the early stage of mammalian development are affected by this Aurora B signal C03 axis. Next slide. So what was really happening? Now we know that Aurora B helps C03 to go to the nucleus, and now we know that if we hamper or tamper this function, there is no differentiation into mesendoderm and endoderm. Now, why? What's going on here? What we also found is these particular mutant cells has high rate of cell death and apoptosis. So then we studied the literature, we brainstormed. So we found that because we were using FGF and BMP, this particular factors acts by ERK pathway. This particular pathway is another very famous pathway. I have been lucky, I've been working with super famous pathways, coincidentally. This is another very important pathway, this ERK pathway, important in cancers and in different systems in the neurons, as well as in the stem cell. So whenever a general cell goes for differentiation, the first thing which triggers up is the ERK pathway. So any like stem cells, whenever it starts lineage commitment, ERK pathway is the one which first starts activating. So I was like hitting jackpot, like, oh, wow. I mean, I found something, ERK pathway was getting affected by C03. Great. So that's what, literally hitting jackpot. It was just like, we were just screening like, okay, maybe this. It worked. So that's what we found. So in this particular mutant cells of C03, you can see the phospho-ERK is vanished. So this particular function, that is Aurora B interacting with C03 and then getting into the nucleus, was somehow activating ERK pathway. And when there is a mutant, that is when the C03 is not able to go in the nucleus, there is something up with the ERK pathway. And then ERK pathway is not active, and the cell was dying, and therefore the lineages were not forming. Next slide. Here you see, sorry, of the double reflections. Here you see another image of proximity ligation assay where we tried to see, this is actually a very good assay compared to immunoprecipitation, because for immunoprecipitation, sometimes you need a lot of cells. And these stem cells are really, really picky. They don't grow well and you don't have enough protein to do immunoprecipitation. So proximity ligation assay was ideal assay for this, where you can see, you can visualize, you can quantify. And was perfect for stem cells. So, which I learned in my PhD lab, I carry over and did the same there. And you can see that in the wild type cells, there is proper interaction of C03 and ERK in the nucleus. You can see number of dots. I mean, there are other quantification and other results as well, which supported our study. But then in the mutant cells, you can see there are hardly any interaction, like few dots, but from the nucleus, it is completely vanished. So we were really very, very happy to see that yes, C03 was indeed interacting with ERK in the wild type cells, in the normal cells in the nucleus. And when it is not in the nucleus, the mutant Bi-Aurora B, it completely messes up the ERK pathway and causes the cells to die. Next slide, please. So to conclude this part of the study, what we see is Aurora B phosphorylates C03, and this phosphorylation helps the C03 to go into the nucleus, where it interacts with ERK pathway and then helps the cell to survive and differentiate into mesendoderm. And when we have the mutant, it can't go to the nucleus and the cell somehow doesn't survive and eventually die. And this has immense implications and further scope of study in regeneration and therapy, which we are still have to explore more. This study was published in Molecular Biology of the Cell. And next slide. So then my professor, Neil Dillon, he retired that time, and it was like COVID time and we couldn't study this part further. And then I currently where I work in and I joined in was Professor Costanza Emanueli. She's the head of British Heart Foundation and cardiovascular professor in National Heart and Lung Institute, Imperial College. And extremely, extremely collaborative, dynamic woman scientist. I'm really happy to work with her. I've been working with her for the past 2 years as a research associate, like as a senior postdoc. And currently, I work in the role of small RNAs. Now I'm currently working in cardiac disease, but also later on to be focused on stem cell and cardiac stem cells. Okay, next slide, please. So here, so now we have moved from cancers. I've been through a very interesting journey and learned a lot of things about biology and moved to very basic stem cell biology. And now here is more of a translational research, understanding the molecular biology of translation, and also then maybe taken further for stem cell therapy as well. So our lab in here in Costanza's group, we are a very big group. We are about 16 to 20 people. 7 postdocs. We study different aspects of cardiovascular disease. She specializes in diabetes and ischemia. And so as you see, nearly 70% of the people with type 2 diabetes show signs of heart function loss, and 5 years of diagnosis. So she's very, very collaborative. She has immense number of collaborators all over the world, and she's really keen on identifying biomarkers, molecular signatures, and how we can help patients in quick diagnosis and also exosome therapy. Next slide. So yeah, ischemic heart disease, very famous, very deadly, and also very well studied and still under a lot of study. So ischemic heart disease is caused by lack of blood flow to the heart and is one of the major causes of deaths worldwide. And type 2 diabetes is a major risk factor for ischemic heart disease, which henceforth I will mention as IHD. It induces microangiopathy, and so angiogenesis is a very, very interesting thing. So in cancers, angiogenesis is a bad guy, okay? Because it induces the blood vessel formation, more cancer cells, and it's bad. But in heart disease, angiogenesis is good. Because in heart disease, angiogenesis helps in repairing the blood cells and increase the circulation where there is a scar tissue formation. So that's why angiogenesis is very reparative in heart disease. And then, of course, there is decline in endothelial functions. So endothelial cells are very important because it forms the lining of the blood vessels and of the heart, and critical in functioning of the heart cells, apart from the cardiac cells. Next slide. So again, coming to the different types of RNAs and RNA binding proteins. So here you see is a cartoon, which is RNA family portraits. Apart from the canonical messenger RNA, transfer RNA, whatever RNA, the regulatory RNAs, which we called, now is of course the long non-coding RNAs are also in place. So there are, apart from the coding RNAs, there are non-coding RNAs. The first non-coding RNA, the famous one, was discovered in the late 90s, LET7 microRNA. Then, there has been very, very good. Also, Shubhendranath Bhattacharya from IICB has been working as one of the pioneers in microRNAs as well. And so there's a lot of different regulatory roles of microRNAs. Apart from microRNAs, there are also recent different types. So now is a very hot topic is long non-coding RNAs. Apart from that, there is also circular RNAs, piRNAs, and different form of novel new classes of RNAs, which doesn't code into genes and proteins. But what they do is regulate other genes and transcriptions. Just to give you an example, like how microRNA works. This microRNA is processed, and then, apart not going to the canonical mRNA functions, it is combined by dicer and the RISC complex, and then there is a particular, it identifies complementary sequence in target genes, it binds the target genes, and then represses the transcription of the target genes. It's a very basic function of microRNAs. Next slide, please. So what I work on is not microRNAs, because our lab has been working for microRNAs for a long time, but the new RNAs is a circular RNAs. It's very fascinating as well. So circular RNAs are closed long non-coding RNAs. So basically it forms by alternative splicing. So it's a linear RNA, but from a particular junction, it excises out and forms the circular RNA. There has been immense functions. They are quite well studied in different pathologies, in cancers, in diabetes, in neuronal systems, in neurodegenerative diseases as well. So the most important function is what it does is the circular RNA goes and sequesters microRNAs. And then it binds the microRNAs so that the microRNA then can't go and repress the target gene. Hence, it acts as a complementary function of the microRNAs. Besides, it can also interact with proteins and other termination factors in elongation and translation as well. Next slide, please. So in our lab, we get direct left ventricle biopsies from the hospital.
Diphtheria cases are also increasing. So since there is increase in diphtheria in parallel to the tetanus also, so wherever we get a chance of immunizing against tetanus, we should immunize against diphtheria also. Therefore, TT standalone vaccine has been taken out from
I'm
the
sorry,
even
Momita,
WHO
just a minute. Let me mute
stopped
the
the
person.
production of this TT, and all the
No
vaccines which
problem.
are
No
available
problem.
are now TD only. So just in a short, just to provide protection against the diphtheria.
You can continue now.
Thank you. So our lab receives biopsies, which is from the left ventricle of the heart, from the hospitals and directly from the patient samples who has ischemic heart disease and diabetes. So what we have done is we have sequenced for small RNAs, microRNAs of all this, and my colleague Dr. Mariam Anwar has analyzed and identified different circular RNA and microRNA networks which are differentially regulated compared to the normal patients in the ischemic patients and patients with ischemia and diabetes. So this project was based on identifying top circular RNAs and microRNAs which are aberrantly regulated and their mechanistic functions in the endothelial cells. Next slide, please. So here, this is a network. It's a very novel. We are about to submit this paper. This is a very novel network where she has created this network. It's the data from my colleague Mariam. So bioinformatically, she has identified and networked a couple of circular RNAs and microRNAs. And we have screened a couple of circular RNAs, and we found this particular circular RNA called NPHP1. It is basically, it is from nephrons, that is from the kidney, but the circular form is more important in the heart. And we were trying to understand what is its function in the heart. You can see below from the graph, this is from the RNA sequencing data, that in the presence of, in the ischemic patients and the diabetic patients, the expression of this particular circular RNA is high in the patient samples. Next slide, please. Then we have also validated by PCR from the samples, and we did see similar results. That is, this particular circular RNA is increased in the patients with ischemia and diabetes. Now, to mimic ischemia and diabetes in the cells, what we do is we culture the cells in hypoxia, that is 0.1% oxygen instead of 20%, and we add high glucose to mimic diabetes. So in vitro conditions, we got the similar results, that is in presence of hypoxia and high glucose conditions, that is ischemia and diabetes, this particular circular RNA is highly elevated. Next slide. And then this is a classic assay of angiogenesis in the heart. So this is called matrigel assay, where we plate the cells in a matrigel and we see the tube formation. That is, these are the tubes which are very beautiful network, which usually the endothelial cells form in the heart. This is an in vitro experiment. So here we have again silenced, that is decreased the efficiency of this particular circular RNA. And we observed that the angiogenesis is highly affected. That is, upon modulation of the circular RNA, the angiogenesis are not effective. As you can see from the graph, also the proliferation of the cells were affected as well. So the cells were not proliferating well, and the angiogenesis was hampered when the circular RNA was tampered. Next slide. So we also fished out what it can affect, which particular microRNA it can affect, and we screened several microRNA as well. And in one of the assays, we pulled down the circular RNA by a RNA pull down experiment. And I found that it was binding to microRNA 2213P, which was known to be very important for angiogenesis. And we indeed find the factors VGF, which is important for angiogenesis, and BCL2 for cell proliferation getting affected by this particular circular RNA. Next slide. So in conclusion, from this project, what we see is this particular circular RNA is upregulated in patient samples, also in vitro in the endothelial cells, in patients with ischemia and diabetes. So what it does, it binds and sequesters all the microRNA so that it can upregulate the expression of VGF, which is important for repair of the heart and heart regeneration and proliferation for proper functioning of the heart. And currently, there are different projects. We also work with methylation and regulation of microRNAs. We are also working on cardiac stem cells repair and other small RNAs like piRNAs and long non-coding RNAs as well. Next slide, please. This is our group. It's pretty large. You still see half of the group, half of them are still not here. That's my professor in between, Costanza, and different bioinformaticians, wet lab people.
So this is
and
which
the
category?
group-
Yeah, this is obviously 3, so everybody is right.
Okay, fine.
The
So
next
this
slide,
is category 3. So leaks on mucous membrane, important. Leaks on mucous membrane or broken skin, bites with
Next slide. And finally, this is the last slide where I would acknowledge definitely Professor Costanza Emanuele and her group, British Heart Foundation for the grants and the money, Professor Neil Dillon and his group, UKRI for the research, and definitely Professor, my PhD supervisor, who AUC basically the Hatekori of your PhD, Dr. Malini Mosu ma'am, and CSIR for my fellowships and for all the support. And thank you all for giving me the opportunity for introduction. And it was great coming and giving talk here and interact with all of you. And I'm happy to answer questions and any kind of general or scientific questions as well. Thank you very much.
Very good presentation and very useful work
Thank you.
with reference to treating several lifestyle diseases. I hope you are overwhelmed and very satisfied, I guess, with your progress.
Well, initially it was a bit overwhelming, but
Mhm.
at the beginning I was like, no, it's, but I think, yeah, after all this time, I think I've learned in depth in several systems and broadened my expertise technically, scientifically.
But when we see pathogenesis, no, your title you said gene regulation in pathogenesis.
Oh, you felt more like a microbial.
Exactly.
Yeah.
So that was one. I hope Dr. Malini will allow me to ask 2 questions, not many. Is it okay?
No, you can ask as many because we have very few people to ask questions.
Yeah, few of the people left also in between.
Yes.
Because in between, some other speaker was coming on our way of this presentation.
No, yeah, that this is the first time I have met with this sort of. Okay, sir, you go forward.
Yeah, no, actually, colon cancer in microbiology, we see more dysbiotic gut, actually, right? Yeah, but you were telling a totally different phenomena with reference to this non-coding RNAs. So, of course, dysbiotic gut actually arises because of this imbalance or disturbance, in other words. So, it is a result of the whatever this gene regulation or function it happens
Yeah.
at molecular level, and then there is a imbalance in the gut dysbiosis. There is
Yes.
gut dysbiosis. But then how actually you link, because no, most of this colon cancer we actually looks to treat through the suppose fecal microbiota transplantations. So will it reverse those changes at the genetic level?
So at the genetic level, because colon cancer, as you know, sir, also because very inflammatory as well. So it's
Exactly.
more for what you say combinatorial therapy, because one thing, colon cancer was highly treated by Wnt signaling, but then there was still relapse because only one particular pathway is more studied. So why it is relapsing? Because it is cross-talking. That was one of the major point of our research, that we used to study crosstalks between other paths, the how it bypasses and activates the other path when one particular drug is blocking one path. So that's what we studied in colon cancer, because most people use drugs for Wnt signaling pathway.
Okay.
But then we could decipher, like at least from our group, and then also other groups are reporting as well. So our group, we deciphered that also STAT pathway is important and NF-kappa B pathway is important. So the combinatorial drugs is very important to help the cancer from relapsing or bypassing or creating chemoresistance for one particular drug.
Exactly. Another thing was this FOXO mechanism regulation, or even NF-kappa B actually modulates even through probiotics. So you
Yes.
think that this also can have a something like an influence for the treatment?
I
through the
think this is a
probiotics?
very good point. This is a very, very good suggestion and point from you. I think if FOXO helps in probiotics, I think treating the patients or adding probiotics might really help also to, improve the treatment. I think this is a very, very impressive point we have raised.
Now, as from the therapeutic purpose, how actually is snRNA will be used in future for a therapeutic agent?
Yes, so that is something a very,
Mhm.
very novel field of study. So you can so first of all, snRNAs can be inhibited as well, like normal genes. People are trying to deliver like lipid nanoparticles to affect the snRNAs as well, but these are currently under trials.
Exactly.
And also, in exosomes, and you can package the small RNAs in lipid nanoparticles but still things are ongoing. So how it is responding and, cross-reactivity, non-specificity, but looks very, very promising.
Wonderful, wonderful. Thank you. Thank you very much.
Thank you so much as well.
Yes, thank you, Momita. Like, it's a huge story, huge in-depth study. But the way you deliver to us, like even for people who are not exactly related to this field, but of course has interest. And so definitely, I hope like it has opened some interesting paths for people in the upcoming years or sessions. So once again, a very warm welcome and thank you once again from the MBSI platform. And thank you, Halami sir, for being from the start to finish, as you always do, and come up with ideas for
Thank you.
interactive
Thank you
things.
very much.
Yes, so it is nice. At least we have some people like devoted listeners and like interactive people to work on, because these are huge things to work on. So, Momita, if you can just quickly run through your messages, a lot of people have appreciated your huge piece of work before we end the meeting.
Yeah, we already spoke to Prakash sir right now.
Yeah,
Yes,
definitely.
yes.
My question you did not repeat. Maybe other things you can just have a look.
No, actually, yeah, there wasn't questions. There were very like appreciation comments rather.
Exactly,
So
exactly.
that's why I'm
definitely.
asking Momita to go through so that she leaves the
Yeah,
meeting with a
definitely.
huge sort of satisfaction. Like she has been known to me for a long time, and I know her devotion to work and her commitment and her nature of interaction, everything. But this, I wanted to percolate to the community so that the new students, the promising students, like they can find interest and work on also. So once again,
Definitely.
like yes, once you come to
Everybody.
India, like, I mean, we will definitely arrange
Definitely.
for one on offline meeting. Now this on and off, this is always going on and off, like we should think second time. So anyway, thank you so much. So see you
Yeah,
again.
run through.
Have a nice time. And I don't know, like, why you appreciated me. It was just a mere connection on my part and nothing else. So,
Maybe for
but
a minute I ran through the messages as well, ma'am, as you requested.
yes, yes,
Messages from the
run through
chat.
messages. You run through the messages. Yes, yes.
Thank you, Dr. Bhavana. Thank you so much. And of course, Prakash sir, we already spoke. Thank you so much. Thank you, Dr. Kirat. Thank you very much. And thank you. Thank you, Purvi ma'am. Thank you,
Yeah,
Supriya.
yeah.
Thank you. Thank you so much. And thanks to all ma'am, of course, for inviting me, and all of you for the interaction and the kind greetings.
Actually,
Thank
this
you,
is an
Vikas and
opportunity which
Vidya.
I could make use of. So I am thankful to the society for so that I know people whom I know, like who has the objective in percolating their work, their notion to others, that I can make use of. Okay, have a nice day. So
You too,
thank
all
you.
of you.
Yeah.
Have a nice Sunday, and bye-bye. See you
Bye.
again, and good luck for everything.
Thank you. Okay, bye.
Thank you. Thank you, everyone.
So, sir, I'm ending the meeting.
Definitely.